Hello, I'm Dr. Tanmoya Nemai Ghosh, and this is my current research presentation on nanomaterial to bioassay optimized by machine learning for non-invasive diagnostic. In this presentation, I will discuss how nanomaterials, bioassay engineering, and machine learning can be integrated into a unified framework to develop robust, non-invasive diagnostic platform with a focus on sensor optimization, biomarker detection, and translation potential. In my present research work, I have developed a point of care diagnostic biochip as shown in the figure, which has the capability of giving results within 10 minutes. And this was developed for early detection of ocular diseases, that is, age related macular degeneration and retinopathy of prematurity. And these are the leading cause of irreversible vision loss. And in these diseases, we can see that the early stages are often asymptomatic, and this makes timely diagnosis very challenging. And the current diagnostic methods such as fundus imaging and optical coherence tomography, these methods are very expensive. First of all, these are clinic dependent. That means a specialized technician is required to decipher the information and to operate the instrument as well, and detect diseases at and this has the flaw that it can disease only when the symptoms come up, so it detects at a later stage. So after knowing all these challenges, we really feel the critical need for cost effective, early, non invasive, and point of care screening tools for these ocular diseases. To develop point of care diagnostic biochip, we first need to fabricate the device. The entire workflow of the uh, device is as shown in this slide. To fabricate a three sensor array as shown, and which has interdigitated micro electrode of 30 micrometer interfinger gap and 50 pairs of IDE each. So that's why our sensor has the capability of detecting three analytes of interest at a time for a particular disease. Next is functional nanomaterial synthesis. We have synthesized 2D nanomaterial vanadium disulfide nanowires and reduced graphene oxide nanosheets. So 2D material provides excellent electron transport between the bioreceptor and our electrode. So, and we have tailored the properties and optimized the synthesis procedure to provide us the excellent materials to be deposited on the top of our electrodes and form a bridge between the two electrode fingers of interdigitated microelectrode. Once the 2D material is deposited in between the two fingers of microelectrode, then we start our biofunctionalization process of 2D nanomaterial. In case of vanadium disulfide nanowires, we have used thiol based linkers, that is mercaptopropionic acid based treatment, and for functionalizing our bioreceptor on the top of it. In case of RGO, we have used P-base and APTS linkers to immobilize the bioreceptors. The ML assisted framework for biochip process optimization is shown, and in that we use two techniques, that is central composite design and ML models to obtain the optimized parameters. Once we obtain those parameters, we prepare the biochip and then we calibrate it against different concentrations of biomarkers. After sensor calibration, the clinical trial for ROP and AMD is done using the hospital samples, tier samples, and given by our hospital collaborators. For ROP sample, we got excellent results with our kit, that is 100% sensitivity, 96.97% specificity. And in case of AMD, we can see that the kit response is correlated with ELISA response, and we get accuracy of about 97.7%. That means this biochip has the capability to go for further clinical trials and thereby for community based screening.